It's now my pleasure to introduce our speakers for today's first sessions, which will be a spotlight on generic drug review challenges and solutions. First, we'll hear from Tina Zhao, a chemist with the Division of Product Quality Assessment 4, Office of Product Quality Assessment 1, within the Office of Pharmaceutical Quality. Then we'll hear from Dr. Maria Flynn, a Senior Pharmaceutical Quality Assessor within the Division of Product Quality Assessment 8, also within the Office of Product Quality Assessment 2 in the Office of Pharmaceutical Quality, and followed by a presentation by Dr. Nashwa El-Gendi, Senior Pharmaceutical Quality Assessor within DPQA 5, and again, the Office of Product Quality Assessment 1 within the Office of Pharmaceutical Quality. All right, and now I'll hand it over to Tina Zhao.
Okay, today I'm going to talk about quality-related review considerations in teriparatide injection first generic approval. So my presentation will cover quality-related recommendations and immunogenicity risk mitigation for teriparatide injection generic applications. I will also go over some examples of common deficiencies for NA peptide products. My presentation will help you to learn quality-related recommendations for teriparatide injection generic approval, and also some common deficiencies to highlight aspects where attention should be focused to facilitate ANDA approval. Teriparatide contains 34 amino acids, is indicated for osteoporosis. The drug product is a clear solution in a multi-dose injector pen for sub-Q injection. The reference product is from Eli Lilly with recombinant API. The first approved generic ANDAs are from Teva and Apotex. Both are Q1, Q2 the same as the RLD, and both are eligible for biowaiver, and both with the synthetic API. For ANDA submitted under 505J, the applicant must demonstrate that the proposed generic drug has therapeutic equivalence with the RLD. A drug that is therapeutically equivalent also needs to be pharmaceutically equivalent and bioequivalent. I want to point out the same active ingredient is required by both PE and BE. For teriparatide injection, FDA just published the product-specific guidance in this August. Besides the Q1, Q2 and the device recommendation, this PSG also direct the applicant to follow this FDA peptide guidance for the quality considerations. In my slides, I also list those peptide products that whose PSG also reference this peptide guidance. I also list those peptide products whose PSG recommend active ingredient sameness. The FDA peptide guidance describe the comparative characterization of the following attributes for the proposed drug product and RLD. Primary sequence and physical chemical properties, secondary structure, oligomer aggregation states, biological activities, and also the use of orthogonal analytical methods is recommended. These slides provide more detail of the active ingredient characterization and comparability studies. To characterize the drug substance primary structure and physical chemical properties, the firm needs to provide evidence to ensure the identity, strength, quality, and the purity of the peptide drug substance. DMF can be used for this AI characterization. For the comparability, the firm needs to provide evidence to ensure the peptide higher order structure, oligomer aggregation profile, and the biological activity in the proposed finished drug product are comparable to those of the RLD. This slide provides the examples of characterization comparability studies at the drug substance level, and also some examples of the analytical methods. To characterize the primary sequence and physical chemical properties, the firm's study needs to include primary peptide structure sequence, solubility as a function of pH, high growth capacity, log P, isoelectric point, specific optical rotation. The analytical method could be IR, MS, UV, NMR, CD, AAA, L and D amino acid content, AAS, Edman degradation, and or high resolution MS. These slides provide the examples of characterization comparability studies at the drug product level and some examples of those analytical methods. For the higher order structure, the far UV-CD, FTIR, Raman can be used to characterize the secondary structure. Near UV-CD, intrinsic fluorescence, DSC NMR can be used to characterize the tertiary structure. For the oligomer aggregation states, the light scattering, analytical ultracentrifugation, gel electrophoresis, FFF, MFI can be used to characterize reversible non-covalent soluble oligomers, and also those irreversible soluble insoluble aggregates. For the biological activities, bioidentity tests per USP monograph for teriparatide injection can be used in the therapeutically relevant bioassay. Moving on to the impurity comparability studies. Per the FDA peptide guidance, the impurity should not be greater in the test product than that in the RLD. The firm needs to conduct a comparative peptide-related impurity profiling of the RLD and the proposed generic product to identify each peptide-related impurity at level greater than or equal to 0.10%, to demonstrate the no new peptide-related impurity greater than 0.5%. For impurity found in both the proposed drug product and the RLD, the firm needs to demonstrate the level in the test product is same as or lower than that of the RLD. For impurity found at level between 0.10 and 0.5%, if not present in the RLD or in the test product at higher level than that of the RLD, the firm needs to provide justification as to why such an impurity does not affect the safety, effectiveness, or potential for immunogenicity. And also the firm should consider to use the UHPLC high-resolution MS to facilitate peak identification and ensure peak purity. Regarding the immunogenicity risk mitigation, for any new or elevated level impurities found at level of between 0.10 and 0.5%, adaptive immunogenicity risks for such impurities should be evaluated individually on the purified impurity standard. Innate immune response evaluation should be conducted on the fully formulated drug product in comparison with the ROD to ensure the generic product does not contain anything such as impurities, aggregates, contaminants, or leachables that may increase immunogenicity risk. Next, I want to go over some common deficiencies for any peptide products. The first example, the firm performed an impurity profile characterization of the test product samples aged near release using the HPLC-related substance method. FDA recommend the firm to update the study using test product samples aged at the end of the proposed shelf life and perform the characterization study using orthogonal methods that use different chromatographic separation principles and also use UHPLC high-resolution MS to facilitate peak identification and matching between the ROD and the proposed the product to characterize peak purity. The next example, the firm identified some impurities in the spec table with RRTs. FDA recommend the firm to characterize the structure of those impurities identified by RRTs as per the ANDA peptide guidance and update the spec table with identified name based on the characterization. The next example, the firm proposed a limit of no more than 0.10% for any unspecified impurity. FDA recommend the firm to revise the limit from no more than 0.10% to less than 0.10% to be consistent with ANDA peptide guidance, which states that all impurities presented at 0.10% or greater should be identified and characterized. The next example, the limit proposed by the firm for impurity 1 is more relaxed than no more than 0.5% and not supported by the comparative RD data. FDA recommend the firm to tighten the limit consistent with and supported by the observed the RD level, or tighten the limit to 0.10 to 0.5% and supported by immunogenicity study results, suggesting the impurity 1 does not add immunogenicity risk to the product at the proposed limit. The next example, the firm reported a greater level of aggregates in the test product than those found in the ROD. The FDA recommend the firm to perform an investigation to identify the root cause and improve the product aggregation profile. The aggregation profile of the test product should be comparable to the ROD. And also the firm needs to demonstrate the capability of manufacturing drug substance or drug product, exhibit batch as necessary with acceptable control of aggregates across the proposed shelf life. Now we're moving on to the challenge question. The question number 1, consistent with the ANDA peptide guidance, when conducting a comparative impurity profiling of the generic product and RLD, identify all peptide-related impurity at a level of A greater than 0.10%, B greater than 0.5%, C greater than or equal to 0.10%, D greater than or equal to 0.1%. The answer is C. Per the FDA peptide guidance, the firm needs to identify all peptide-related impurity at a level of greater than or equal to 0.10%. Challenge question number 2. Which of the following statements is not true? A, ICH Q3A and Q3B guideline is generally applicable to all drug products, including peptide drug products. B, the limit for each peptide-related impurity can be qualified by the observed RLD level for the same impurity. C, a comparative characterization of the impurity profile of the generic product and RLD is recommended to establish acceptance impurity limits. D, peptide-related impurity and aggregates may pose immunogenicity risks. The answer is A. Per the ICH Q3A and Q3B, the peptide drug product are not in the scope of ICH Q3A and Q3B. In summary, my presentation provided the overview of the quality-related recommendations relevant to teriparatide injection generic approval, and also provide some, we go over some examples of common deficiencies for those, for any peptide products. Lastly, I want to thank Cameron, Ely, and Bing for their help on my presentation. That's it. And next, please welcome Dr. Flynn.
Good morning. I'm Maria Flynn, and I have a question for you. Do you know someone who has not been able to afford their prescribed medications? You likely do, even if you don't know, because 1 out of every 3 Americans can't afford their medicines. In fact, if they could afford them, they may not be able to afford the most advanced forms. First generic drug approvals bring down the cost of medications. Today, I'm going to be referring to the first generic oral liquids, and those approvals actually done just recently. Oral liquids are very important to vulnerable populations, such as our geriatric populations, our elderly or wise old friends, as well as the youngest, the pediatric populations, and other patients who have swallowability issues. So today, we're going to have oh, there we go. He told me to look panic. So our learning objectives today is we're going to discuss the quality attributes for oral liquids. And we're going to do this in the context and the framework of 2023 approved applications. And so we're going to be discussing 4 case studies. The first 2 case studies, we're going to be discussing the stability of an active pharmaceutical ingredient, also known as an API, or actives. And so the very first case scenario, we're going to be discussing physical stability. And when we think about physical stability, we're actually thinking about the structure of the active. In the second case scenario, we're going to be talking about the chemical reactivity of that active. So if it can degrade, something like that. And in the third case, we're going to be discussing the unexpected leachable situation, as well as our final, which is going to be a container closure risk management scenario. So this slide is showing all the possibles, well, not all, some of the critical quality attributes. You always have to leave room for that. And moving forward, I'm going to be discussing very specifically in the case studies, I'm going to be specifying if it's an oral solution or oral suspension. So when you think about oral liquids, I want to refer you to a chapter in USP. And there's a chapter 1151 that goes through all the different dosage forms for oral liquids. And in that, they'll define each one. So I will not be discussing today the other dosage forms, such as a powder for solution or suspension or syrups, because the last first approvals from 2023 were oral solutions and oral suspensions. So in thinking about the critical quality attributes, we already discussed stability. And why is that so? So when you're in a liquid phase, unlike your tablet where you have your active that's in a fixed position, you have fluidity. You can actually see many things because you're a liquid. So if I were an active, I could see other actives. I can also see other excipients. I could see solvent and all the permeations above. So because of that, they are more reactive. And a lot of times, you will see that you have to have different storage conditions. They might have shorter shelf lives, and they might have some instability. And so we talked about physical stability and chemical stability. And we were looking at assay. We assay the active. And sometimes the active, you can actually see if it is degrading, you can see changes in the assay. But there are also other functional parts of the formulation that we are going to assay as well. Content and uniformity is applicable, but I want to even throw out there another term for oral suspensions, and that's going to be dose uniformity. And so this is very important when you think about suspensions. All throughout the bottle, a lot of the cases, they'll tell you to shake it. And all throughout, you want to make sure from the top, middle, and bottom that you get a homogeneous dose given to the patient. And also over its shelf life, you want to look at dose uniformity. Container closure systems are very important for liquids because they have to be airtight. You don't want to have leakage or spillage. Dosing accuracy is critical in making sure you get the right dose to the right patient for the right indication. Microbial limits. You're in a liquid, so there's water activity that can promote microbial growth. So microbial contamination as well as controls need to be in place. And a lot of times we'll see preservatives being used to be antimicrobial. Leachables are relative to our solid oral dosage forms, they're of higher risk. However, maybe not as much risk as we saw in TNUS when you're dealing with injectables. Dissolution, we're going to reserve to oral suspensions because the active's being suspended, and so you care about it being into how it goes into solution. So let's go ahead and dive into our first case study. When we're thinking about physical stability of oral solutions and suspensions, we're thinking about the solid state. And this is for both dosage forms. You're thinking about the different forms that can occur. The last two, sedimentation and particle size growth, this is only reserved for oral suspensions in that that's what, when you have to shake it, it can either sediment or there could be particle size growth over time. And you need to control that again for dose uniformity. So those we'll reserve. But I want to also throw out there another comment about redispersibility and resuspendability throughout the shelf life in each dose. So with physical stability of oral solution and liquids, we think about solid state, and we can ask ourselves some questions, for instance, about the active. Is it known to have polymorphism? If it does, there might be an increased risk. We can ask, is the form crystalline? Is it amorphous? Suspensions can have a much greater risk than solutions because in suspension is thermodynamically unstable. So inherently, it can have more risk. But whenever you're thinking about different forms, you really need to think about the solution equilibrium of each form. So when we're thinking about solid states of the oral solution, you can think of it lower risk because it's all dissolved if it's a true solution. And if you know that your active is dissolved in a concentration that's less than the solution equilibrium, you can think about a lower risk. Now, for oral suspensions, while we mentioned that they're at higher risk, there's some things that you can think of initially that you can think that it may not have as much risk. For instance, if there's no polymorphisms, it's not existing, you don't see it, then, and it's not in the literature, and so far under your control, you don't see it, it may not be a risk. Or you're using the most stable form. And then there's times when all you know is the amorphous form, and in that case, there's a likelihood that there wouldn't be a crystalline form. So that can initially lower your risk. But when we also think about actives, we think about them in terms of solubility. And with solubility, we think about the high solubility class, and we use a class system called BCS, which is the Biopharmaceutical Classification System, and it explains what would be considered highly soluble. So if your active is highly soluble, it's more likely it's not going to have risk. Now, you can increase the risk if you know that you're using the unstable form. And if you also have multiple forms and you know they interconvert, that can also increase your risk. So let's dive into our first case study, and we're dealing with an oral suspension. And this oral suspension is known to have polymorphism. You can see the different forms. It's crystalline, but again, as I mentioned, the active is being suspended, okay? And so there's a greater risk thermodynamically for that. It happens to also be in a risk category of poor solubility, a BCS class 2. So what do they do? What do we do in this situation? Well, you need to mitigate it. Before you even go into formulation and development, all that wonderful things, you need to control your drug substance. You want to make sure you know the polymorphic identification. You want to have a 3-tier particle size for the drug substance. This is before you even get to the drug product. Then, once you know that, you go through manufacturing. Things can happen during manufacturing. In the very end, you get your drug product. So at release, you want to make sure that that polymorphism is what you have. It has not changed. But also, over the shelf life, you want to make sure that it remains in that form. Now, in the drug product also, you want to make sure you have a 3-tier particle size for the actual suspension, that you know what exactly the particle size, and there's a range, and there's 3 tiers. Now, I show you a number that's less than 22 microns, but there are 3 different numbers that make that up. I wanted to put that number there specifically because I wanted to show that this is highly micronized. And this is a trick that pharmaceutical scientists do. We love to, I don't know if I can call myself a pharmaceutical scientist, but I'll put myself in the category since I have 16 years experience. We love to have very highly micronized compounds because you increase the surface area, which then increases your dissolution rate. For pharmaceutical, so that's your going into and what you know. But furthermore, since we knew that this fit into such a high risk category, the applicant for this first generic showed a polymorphic study. And they showed that at the 6-month accelerated, as well as at the 24-month, the long term, which happened to be the shelf life for their drug product, there was no conversion. So they had a good control, and they're going to keep all these controls in place and watch it, and so make sure that this does not happen and that they have the form, because it's very important. So now diving into chemical stability, we're going to be talking about different risk. Usually by the time an applicant comes to FDA, they have at least 3 months, usually 6 months of accelerated as well as long-term data. And at that first initial risk, you can either see that there's no trend, or you can see significant trend. If you don't see trend, initially, it might have a low risk. That doesn't necessarily mean that will happen over the course of the shelf life, but you can feel a little bit sense of security. When it comes to risk, when you see significant trends, so if you see the assay decreasing, for instance, or you see other degradants forming over time, these significant trends, and especially if they go out of spec, really give you a heads up that there's something unstable about that formulation. It could be obviously we're looking at the active in this particular situation. But there's also tricks that we do in pharmaceutical science that actually stabilize. And so if you see stabilizing agents, for instance, specifically for the API, if you see that going on, then it might give you a heads up that there's a higher risk. So this case study is talking about the API degrading. Even though we're talking about oral suspension here, I can't tell you how many times this come across our desk, where this is universal to all dosage forms. So I put the label here specifically. Sometimes you can get some hints in the RLD labeling about what possibly can go awry, or what can be the failure modes for your formulation or their formulation. So in this particular case, with much evidence I'm not going to show here, the API was thermally unstable, so it was affected by heat. So when you look at the stability conditions, how they knew that this was happening in their specific situation is that they saw at the 6-month accelerated, and I want to mention here, this is a refrigerated suspension. And so the accelerated is at 25 degrees. And they actually saw a significant trend in the assay. And when we say significant, it means greater than 5%. And they also saw out-of-spec degradants. In the long term, the refrigerated, everything was fine. It complied with all the criteria. So the problem is, if you read our guidance the guidance for ICH-Q1E, for refrigerated compounds, it's very important that it be supported by real-life data. And so in this case, they wanted 24 months stability or shelf life. They wanted that. And so they needed to back that up with real science. And there's a little bit more going on for the suspension. And so it was very important to see that they had a stable formulation, formula. So we're moving on to our first challenge question. And I want to, sometimes we read these quickly, I want to put your eye in the lens of the API. What is not an active pharmaceutical API-related risk factor for oral liquids? So A, exhibits polymorphism, B, dosing cup inaccuracy, C, poor solubility, D, easily oxidized, air sensitive. So I'm going to give you a couple seconds to think about that. So the answer is B. Dosing cup inaccuracy is a risk. It's just not related to the API. The other three can be failure modes for actives. So moving on, we're going to discuss a case 3 study where you're seeing an out-of-spec degradant. This one's very interesting because only one batch of the 3 exhibit batches failed at 6 months accelerated and at the 24-month long term. In fact, the degradant that they didn't even know they had popped up and started to exceed the limits of ICH-Q3B. I want to refer back to Tina. She had a special case when it came to peptides. They had a much stringent, in this particular situation, they didn't have anything very special about their active, and there was no cohort of concern, but they were able to use the guidance ICH-Q3B for their drug product. So this degradant it actually exceeded the identification threshold criteria. So under the identification threshold, you don't really need to know, you might want to know, but you don't really need to know what is there because it's considered safe. So what's going on here? They had to figure out what this was because they now need to find out why this one batch had this degradant. And so they identified and characterized, and imagine, this is a first generic approval. So there's no standards. So they needed to confirm this. They do all the wonderful organic chemistry, and they do I'm actually an organic chemist, and so they actually did the structural elucidification, but to really confirm, confirmation of this is the synthetic, actually synthesizing this. So what did they have? Well, they found out it was very similar to an oxidative degradant they already knew about, but it was a diastereomer, which means one of the chiral centers was racemide. And thinking as an organic chemist about how this can happen, it wasn't just like an acid, like the pH of the oral liquid or suspension. Oh, this is actually an oral solution. In that situation, they couldn't really think about how this could really happen. So they had to think a little more. Why did that one batch out of the 3 fail? What happened? What changed? What was different? So they did their homework. What they learned was that one batch, after much thinking, that they were using one normal HCl to adjust at the very end that drug product formulation. And the difference there was the one normal HCl sat in a stainless steel vessel for longer than the other 2 batches. They found that there was iron being leached out of the stainless steel vessel into this acid that is now going to be used for the drug product formulation. And an oxidized reaction catalyzed by the iron can make that degradant. And they went along and did the science and showed that they did have iron that was a higher concentration than the other ones. And they also realistically, theoretically, it made sense. So that explained why this degradant was growing over time and that why they failed stability. So with that in mind, they had a control strategy. They revised their specifications, and they now are controlling this degradant, and they controlled it at the ICHQ3B. Again, they're allowed to do that. And they committed to even further to do this, since now they understood it, that they would go ahead and they would control this at even a lower limit on commercialization. So what did they do well? They submitted a well-written CAPA, a corrective action preventative action report. They understood the origin of the degradant. They confirmed it by synthesis, and they used a robust analytical methodation to actually see it. And not only that, they committed and they will specify the degradant at a tighter limit, not that we had asked them to do that. They chose to do that. And they got first generic approval. So here's our second question, and I know we're short on time, so I'm going to move quickly through this. All are common deficiencies for oral liquids. So what is a common deficiency? Would it be a well-written CAPA report with a control strategy for unexpected finding? Is there an incomplete understanding of the manufacturing process? Is there not specifying the container closure description and integrity? So if you think about this, what is a common deficiency? You know the last 3 are common deficiencies, but a well-written CAPA and understanding an unexpected finding, that's a good thing. So all the other ones would be common deficiencies. So moving right on, now you reversed you're doing a first generic and you reverse the RLD, reverse engineering, you know what's going on, everything's so wonderful. You made this wonderful formulation, and then you choose the wrong container closure system. So we want you always to look at the packaging evaluation and make sure that even over the shelf life that you are not having any problems with your different configurations. Because in our first case, and our last case, we're going to be discussing this particular picture here, which is 1,000 words. This seal was not appropriate for this single-use unit. And what happened, this is very bad for the patient, they're not going to get the right amount of dose. But furthermore, this could be a hazard for the healthcare professionals and just a mess, depending on what the drug product is. So this was withdrawn. And this first generic approval for the multi-dose containers that had a good seal, they were fine, and they were able to get first generic approval. However, this one is not acceptable. And this is inverted, so when we do these liquid studies, we do the upright, and then we do the worst case. So it could be horizontal, or it could be inverted. In this particular case, with the single unit dose, it was inverted. So in sum, we discussed the quality attributes and the risk of oral suspensions and liquids. We also discussed risk mitigation possibilities and how they were actually done for 4 case studies in real life for first generic approvals. So I have some closing thoughts. Know your drug product, understand it. Think about the failure modes and the risk. come up with a mitigation strategy. But when all that fails, expect the unexpected, because the unknowns will occur. But when they do occur, really find the root cause analysis, really do the CAPA, and submit that with a very clear control strategy of how you're going to go forward so that you can get a first generic approval. So now I'd like to welcome up Nashwa.
Good morning, everyone. My name is Nashwa Al-Gindi, and my presentation topic today is about quality consideration for first generic tiotropium bromide capsule-based dry powder inhalers. This presentation will deliver several learning objectives. We will explore the general constraints in dry powder inhalation, formulation, and manufacturing. We'll discuss potential development challenges for capsule-based dry powder inhalers to improve the quality and the performance of these products. Then I will share valuable recommendations for highlighted product characterization studies, and we'll end it with focusing on general quality considerations for primary drug product stability batches. First, I would like to give you a brief introductory about DPIs. There is a lot of many classification for DPIs. However, here I will focus on one classification based on the device constituent part, the dose container. DPIs can be meter dose inhaler, which a powder reservoir contain multiple units in one container, or pre-meter dose inhalers, which are plasters, discus, cartridge that contain multi-unit dose inhalers, or capsule, which is single unit dose inhaler, and this is the focus for our talk. As a fact, generic DPI can be more complex to review compared to meter dose inhalers. Also, plasters and capsule DPIs-based DPIs are the main type submitted to FDA since 2015. So the reference listed product, the RLD for the first generic capsule-based inhaler is Priva Inhaler, NDA 021395 by Boehringer Pharmaceuticals that has been approved in 2004. The first generic Spriva inhaler is Loop Inhaler by Lupin and 211287 that has been approved in 2023. Each capsule of this drug product contains a dry powder consisting of 18 microgram of the API, tiotropium, blended with the carrier lactose monohydrate. There is the product-specific guidance, draft product-specific guidance with this drug product has been published in 2023 and includes many recommendations for BE studies. One of these recommendations is that the test and the reference product recommended to be Q1, Q2 the same. Also, the test and the reference product, the device should be similar in the mechanism, the breath-actuated pre-meter single unit dose capsule-based format. Also, the number of the doses, the size and the shape and the resistance, the external operating principles and the critical design attributes. There are in vivo PK and comparative clinical endpoints recommendation in this PSG. However, I will focus my talk here about detailing about the in vitro BE studies. For the in vitro BE studies, should be conducted on 3 exhibit batches, 10 units per batch for the test and reference product. And just here, the table summarizes the 2 in vitro studies for in the PSG for this drug product, the single actuation content with the SEC and the aerodynamic particle size distribution APSD. And here in the second column, the design for each study and the summary of the BE analysis. Here they are very related to the quality tests, the SEC, similar to the dose content, delivered dose uniformity and APSD from quality perspective. Then I will jump here into the composition of the capsule-based DPIs. So usually the composition table for sure, it's usually submitted in the module 3 in P1, which contain the ingredient, quality standard, function, quality, quantity, milligram per unit, and the concentration % weight per weight. Firstly, the API here, the quantity should be per the RLD labeling, and this is the DPI strength, which is the amount of the meter, the amount of the drug from the capsule, not the target delivered dose, which is the mass of the drug delivered from the mouthpiece. Then the carrier, and it should be here based on the PSG for this specific drug, it should be Q1, Q2 the same, and this is the most preferred for most complex product. Then the capsule size and composition, and the capsule shell material may be in some cases different between the RLD and the test, and it may be acceptable or may be adequate in some case. Here in our situation, the listed product is gelatin material, however, the first approved is HPMC material. They are different in moisture content. However, it could be acceptable to use different material, provided adequate justification with supporting data. For example, study to demonstrate that the fragments generated by piercing and actuation of the rupturing of the capsule are not inhaled by the patient. Also, I'm sorry, here also the ink composition should be listed in the composition table. Then I will start with one of the first topic from my presentation about the learning objectives, which is the general limitations for DPIs in general. There is a lot of constraints. Firstly, when we look at the formulation design, as we said before, Q1 the same. Q2, this is preferred. For this specific drug product, yes, it's in the PSG, highly recommended. However, for some applicants, they may come up with different Q2. In this case, rationale and adequate justification should be provided, as well as pharmaceutical development data for in vitro testing or studies for a combination of the between different ratios of the drug to excipient, which including combination above and below these ratios for the RLD and test. Justification is really important here, and data. Also, average. Generally, it discouraged per ICHQ-8 guidance. However, if it's used, justification should be also provided. For example, drug loss during proposed manufacturing process. Another thing is the manufacturability, and for sure, this is powder. Powder characteristics are very challenging. So many powder should be tolerated, should have a tolerated particle engineering. The shape and the surface characteristics of the powder can affect the aerosol performance and the airflow, and as well as the drug release. Another constraint for the DPI, specifically for the capsule-based, is the device. And as I said, per the PSG, the device design is very important to be similar with the RLD design. Also equivalent design, also the operating principles, and the resistance. This is very critical attribute for the device. Now, my first challenging question, to keep you in the loop, which of the following examples can justify the proposed average in DPI? A, drug loss during proposed manufacturing process, drug loss due to degradation of the drug substance, drug loss due to deposition on the device constituent part through unit life. The answer is A. Now I'll go deeper into the challenging in developing generic DPIs. To develop a generic DPI with a consistent DPI performance, which represented by the delivered dose uniformity, for example, and APSD. A lot of unique challenges are related to the drug product formulation, API and excipient, manufacturing process, device design characteristic, and ended with the user interface considerations, which is the patient handling. Now, a couple of examples about the formulation development challenges. Firstly, the risk may come from recrystallization of the micronized material, which could lead to particle growth, uncontrolled particle growth during shelf life of the product, thereby affecting the product quality attributes, specifically APSD and DDU. And APSD and DDU are considered the key performance characteristics and measures in assessing BE studies. And accordingly, it also in turn may affect the in vivo performance of the drug product. Also, missing proper control on critical material attributes of the drug constituent parts of this combination product, like the API or the carriers, without proper justification. One of the big challenge here is the amorphous content of micronized particles. Okay, mitigation. What is the control strategy to mitigate this risk? Include equilibration period or condition to allow the recrystallization to occur prior to any additional manufacturing unit operations. Also, consistency of the manufacturing process. Also, establish particle size distribution specification based on the actual data from the micronized lots used in the manufacturing of the drug product exhibit batch or primary stability batches, and ended with continuous in-use control, which is the routine control of the critical material attributes. Another risk here, based on the diagram I can show you here, this is, sorry. So based on this diagram, you can see the powders, the DPIs consisting of API particles, very small, usually micronized within 5 micron, attach it to the surface of the carrier to form agglomerates. This agglomerate fluidized to form a separate agglomerates once it's subjected to the airflow with the passive DPI, with the inhalation by the patient. It starts to be de-agglomerated to disperse the API particles out of the surface of the carrier to be inhaled into the site of action. So the carrier here is very critical component. So the carrier grade may lead to particle size growth over time during storage. Also, the carrier morphology, shape, and surface roughness may affect the adhesion force between the API particles and the carrier particles and affect the aerosol performance. Mitigation control strategy here is to adjust the ratio of the fine to coarse grade particles if they are there, because if the fine particles can be attached to the coarse particles and may affect the attachment or the adhesion of the API particles. Also, continuous in-house control for particle size and morphology. I will jump here into manufacturing challenges. After formulation, we go into the another challenge with the manufacturing, and I will focus in 2 challenges. Firstly, the blend uniformity. Blend uniformity, because this drug product, the challenge here is the excipient over 90% of the whole formulation. So there is a low drug load. Also the flowability due to excipient lot-to-lot variability. How to mitigate these risks with the control strategy? Firstly, how to select the proper blender type, the equipment used. Control the critical process parameters like the blending time, speed, blender fill level. Also the excipient critical material attribute. Control the environmental conditions, conditioning, hold, equilibration period after blending. In addition to the final blend characterization, which includes the uniformity, bulk density, and particle size. Powder fill, another challenging during manufacturing. Fill weight variation and segregation of the API and the excipient lead to failure in the APSD, GDU, and content uniformity. Also, the moisture content, very critical for affecting the APSD and GDU. Mitigation control risk here. It's important to identify the critical process parameters and characterization of the fill weight, content uniformity, moisture, APSD, DDU, and this is from the filled units through stratified sampling during development. Highly recommended. My last challenge here about the container cluster, which is the big deal for combination products. In this specific product, capsule-based product, it has the device, it has the capsule that containing the powder, and the plasters, which is the primary packaging. Here I will give you some recommendations important for this device combination. Firstly, for the capsule dimension is critical for the adequate insertion of the capsule into the inhalation device, piercing, and drug delivery. So it's recommended to include the capsule dimension in the drug product specification, which is the lock length. What about the blister foil and the overwrap pouch of the the device. They provide the protection from moisture permeation to the blend formulation at use and in use and shelf life. So it's recommended that the functional layers of the foil should be controlled for the thickness of the individual functional layer, that to assure the consistency, the consistent quality and performance of the primary and secondary packaging. Also for the device, the inhaler device specification should include the critical functional parameters, and I listed here a couple of these functional parameters to be included in the specification. Also, another recommendations. For the needle diameter, this device has a needle and a mesh which used for inhalation of the dispersing of the powder out of capsules. We recommend the applicant to investigate the potential effect of the needle diameter and the mesh material on the product performance and powder residue in the capsule after testing APSD and DDU. For the button push in force, it's recommended to be determined over the lifetime of the inhaler containing the capsule in the chamber for consistency. And in some cases, some applicants like to also do the same testing without capsule to see the differences. The cap opening force, which is also critical, especially for patient handling. It's important to demonstrate if this force changes over the use of the unit, of the use of the device. And if there is if so, these changes, whatever it's reducing change or reduced force, is sufficient to keep the cap closed and open by the patient easily. My second challenging question: which of the followings are considered formulation development challenge in DPI? Fill weight, B, excipient grade, C, device resistance, D, amorphous content of sprayed APIs. The answer is B and D. Now, my third topic here is about the drug product characterization studies. Per the revised draft FDA guidance on MDIs and DPIs that has been published in 2018, there are a couple of characterization studies should be conducted on the DPIs as a one-time study to demonstrate the robustness and the performance of the product and to support the labeling instruction. Here in the right hand side, you can see I listed the characterization study that can be conducted, should be conducted for capsule-based DPIs. In general, DPI products units used for the characterization studies should be stored under conditions consistent with the storage conditions identified in the label. Also, to support the proposed shelf life at the intended storage condition, it's recommended that some characterization studies should be performed at the beginning and near to the end of the shelf life of the product's units, including in-use period, cleaning instructions, and robustness. Now I have some recommendation for a couple of characterization studies. Firstly, for the robustness, this is based on this specific product capsule-based DPIs. For robustness, it's recommended to assess the physical robustness of the capsules following the dropping and or vibration of the blister card for any damages. In the cleaning instruction study, it's also recommended to determine the drug residual at the end of the experiment for each unit, in addition to visual inspection for any capsule fragments and particle residuals. Fragment study. This is really critical for capsule-based DPIs, especially if the applicant is using material different from that of the RLD. So assess the puncturing and fragmentation behavior of the proposed capsule in comparison to the capsule of the RLD. Here is in this case, it's HPMC versus gelatin. Even if the applicant using gelatin, it's recommended to still perform this study to confirm there is no any fragments that can be inhaled by the patient. My last recommendation for effect of use study, where we are recommending the applicant to inspect the partially used product units after clinical, coming from clinical studies for any external damages. In addition, to evaluate the functionality and piercing mechanism of the clinical return inhalers for any mechanical defects or failures. Like, for example, the lack of rattling of the capsule, how to pierce the capsule, failure of pressing the button. This is really critical functional defects. My last challenging question here. Which of the following drug product characterization studies should be conducted on the DPI product units at the end of near the end of shelf life? A, cleaning instruction, B, temperature cycling, C, robustness, D, in-use period. The answer, A, C, and D. I would like to end my talk with a general quality considerations for the primary DPI stability batches. Firstly, for the drug product constituent parts, the API, excipients, and device. It's highly recommended to use 3 different lots to manufacture the 3 primary stability batches. Drug substance discrete lots, the device constituent part components, as well as the critical excipients. And here, this is the carrier. For the batch size, given the complexity and the potential risk involved in scaling up the manufacturing process for DPI products, it's highly recommended to submit stability data from 3 exhibit batches at the proposed production scale. However, if this information is not available at the time of submission, it may be acceptable to provide at minimum 1 batch at the proposed scale and commercial scale and to 1/3 of the commercial scale. However, if this is the case, it's highly recommended for the applicant to provide in-process and batch analysis data from several batches that's manufactured using the commercial proposed manufacturing process, like engineering, like developmental batches, performance qualification batches, to confirm the robustness of the proposed manufacturing process. At last, the product characterization studies should be conducted on the to-be-market configuration and version of DPI products, also 3 batches with reasonable sample unit to support the reliability, reproducibility of the product quality and performance. And here are the resources that can help with the DPI products: the FDA guidance, USP Chapter 5, and USP Chapter 601. At the end, my closing remarks here, again, DPI products can be more complex to review than MDIs and other complex products. So thorough characterize and understand the quality of the batches used in your exhibit batches, stability, and also BE studies and everything. Quickly identify the root cause for inconsistent DPI performance and optimize product design at early phase of the development to minimize the risk. Also establish control strategy based on the evidence, scientific evidence gathered from thorough characterization of the stability batches and BE batches. At the end, I would like to acknowledge my office and my division, Division 5, my division director, Yi Ting, Deval Gaglani, my supervisor, and my colleagues in Unit 2. Also, I would like to acknowledge Office of Pharmaceutical Quality Research 2 in St. Louis, Jason, Rogers, Chining, and Nicholas. Office of Process and Manufacturing Assessment, Charmesta, Chining, Chu, and Vicky, and also OGD, Office of Research and Standard, Division of Therapeutic Performance 1, past and present members of nasal and inhalation product team. Thank you so much, and I'm looking forward for all questions. And I'm welcoming my other colleagues, my other speakers, and the panelists. Thank you.
All right, thank you again to our presenters for their fantastic presentations. We'll now begin our question and answer panel to include all presenters and additional panelists joining as well, which I'll introduce in a moment. But in the meantime, we want to go ahead and invite attendees in the room. Please line up at the mics if you have questions. Must have some questions after those really informative sessions, right? Yeah, okay. So as I mentioned, we'll have all our presenters here, and also joining the panel, we have Dr. Yili Lee, with Senior Pharmaceutical Quality Assessor with the Division of Product Quality Assessment 11, within the Office of Product Quality Assessment 2. And also Dr. Brian Newman, a lead pharmacologist within the Division of Therapeutic Performance 1 in the Office of Research Standards and Standards within the Office of Generic Drugs. And also for those logged in online, if you haven't had a chance to submit your questions using the online or on-screen link and QR code, please do so now. And we'll answer as many questions as time allows. So let's go to our first in-room question here.
Thank you for the presentation. So for some of the peptide product that's approved way before the guidance is published, they may not necessarily have any unspecified impurities at 0.10% level as the specification. So would that play a factor in agencies' review when looking at the ANDA applications? Because if they can be allowed to be higher than that, but then we are holding to a much tighter spec and the golden 0.10 number.
So yeah, you're talking about if you're talking about peptides. So for those peptides covered by the peptide guidance, yeah, usually we ask you kind of like identify everything at or about 0.10% and identify control in the specifications. Also, there are some peptides not covered by the guidance, but however, if you look for PSG, the PSG will clearly state like this guidance applies to this product. We have similar expectation for the approval at the end for that type of product. Do you have any question for a particular product or just general questions for the?
Actually, if you don't mind, I'm going to jump in here, having a little bit of work, not as much as you, Lee, in the peptide arena. What we really look for peptides, while that's the guidance of 0.10, when it comes time to really doing the actual work and drug formulation, it's always the comparative analysis versus the RLD. So if you do your homework really well in P2, you're looking at the impurity profile of the RLD. And so if you see the RLD is having a higher limit than you would normally have according to the guidance, you can submit those 3 batches relative to the 3 batches that you have. And usually at expiration, you want to be doing that on the RLD, and you want to be doing that as far along as you can for yours as well. And you can provide a good justification, in which case the agency will consider that.
Great, thank you for those responses. We'll go ahead and move on to the next question in the room, over on this side.
Hi, thank you for the presentations. I just want to note that the presentations that give challenges and potential solutions are extremely helpful to industry. And my question is related to some of the particle size data that goes into the applications. You know, we include all of these particle size testing data, and in module 2, where we do summaries, the agency has published some summary tables for doing particle size by MDRS, and I'm wondering if for particle size done by different methods such as laser diffraction, are you also looking to have us generate some type of summary tables for module 2, or do you not care?
Okay, I answer in general. So in general, everything in the application has been shown and we should review everything.
Right.
So anything included in the P2 summary, yes, we look at it and review it. Any justification provided, anything we usually do. And if we have any concerns, we cite the deficiencies to the applicant for more clarifications. That's in general. Did that answer your question?
I'm talking about the module 2, like 271. I'm just wondering if you want summary tables for particle size along with what's in the module 3.
Go ahead. Yeah, I think if it's helpful, definitely do that. I don't think it's a requirement, but if it's something that's going to help the justification, then you can do that.
And I may add here, I recommend everyone. So whenever you submit anything, you may think about, okay, it will be shown, or it's not important. Provide a reviewer guide. Reviewer guide is really amazing that you can put all the documents with hyperlinks and includes everything you need to. So we usually use this as a very guide for us, help us a lot to find whatever we need. And summaries and tables, that helps also a lot. That's in general.
Okay, thank you.
Again, we'll go on to our next in-room question on this side.
Yes, thank you. My name is Michael Lichminat. I just wanted to say thank you, FDA and CEBA, for having this training today. It's really informative and just gives the industry a better understanding of what the recommendations and expectations are. Just quickly, could be directly to Maria Flynn, Mrs. Maria Flynn, or just in broad for the FDA panel. I just wanted to ask, is there any agency updates to the IIG database, specifically to improve excipient toxicity level in pediatric patient within the oral suspension type, maybe oral suspension specifically dosage formulation, or in general?
I love the question. Thank you so much. So something I didn't have time to go over a lot of times with taste masking and a lot of the different formulations, like for suspensions, there's a lot of intricacies that can occur, and in each excipient can have different impurity profiles. And so he was talking about the IgG, which is the inactive ingredient database. And so there's a database, and you have to look at it not only for the actual exposure, but you have to think about it as the combined exposure of your different excipients. And specifically, in a lot of the excipients that are used, there's going to be trace amount of impurities, and we have a good sense of what those are. And it's really important to know your excipient impurity profile, because some of them are of a cohort of concern. They do have toxicological. So to your end, the database is as current as possible. So you do that, but when you do it from a holistic perspective, because there's going to be multiple excipients. And sometimes when you look at them, like for instance, flavoring, it's very important to get that information from whatever vendor that you're doing, like Tutti Frutti or something. For instance, there's a case today I'm thinking of in my mind where there was an issue with benzyl aldehyde, which is a cohort of concern. And it was in multiple excipients, not excipients, but ingredients of the flavor. And had they not looked at that profile, they would not have known that it exceeded what was normal and healthy and safe for use. So to your end, the database is as good as we have right now. And I believe they updated it. If you obviously, if there's something that's more helpful, and if you need to, please reach out to the agency so that we can include the information that you have or information that you're concerned with. And specifically, if you have something that you're trying to do and you're concerned about it, go ahead and submit those control correspondences to us. And if necessary, not in oral liquid situations usually, but in the more complicated situations, you want to use our pre-ANDA. Make sure you go through that pre-ANDA process if there's something really interesting and fun going on. And the sooner you communicate with us, the sooner we can help you and be proactive.
For sure, thank you.
Fantastic. We have another question here on this side of the room.
Okay, so the impurities above 0.1% should be ID'd for peptide, right? But to what extent the mass spec should, like the identification should be? Do you expect like full structure and sequence information, or can we just do a mass spec comparison and match to the ROD impurities?
Oh, yes. So that's a good question. We go back about the impurities. The general idea is, we encourage you identify everything at or about 0%, and then, do extensive comparison with the ROD. In that way, we understand what's the differences between your ANDA and ROD. If you identify I think for certain impurities, only retention time you say, the same as ROD impurity, that's not sufficient. So we usually ask you to provide further characterization. For example, you say the same molecular weight or like MS fragmentation, the same as ROD. Then if you observe the ROD level is higher than your end product, then that means your limiter is qualified. However, if you observe the level in your product is higher than the ROD, then you may probably think about like immunogenicity mitigation.
Thank you.
All right, if we don't have questions in the room at the moment, we'll move to an online question, but I still want to encourage everyone here, don't be shy, still feel free to come up to a mic if a question pops in your head. So we'll go to our first online question.
All right, this is for Dr. Nashwa El-Gendi. How do you deal with polymorphisms in DPIs? Yes, that's correct.
DPIs in general. Mm-hmm. So for DPIs, this is a powder. So powder characteristic is very critical here. For the API and the carrier polymorphism for the API is very critical, and applicant should show if there is any polymorphism, they should have a testing for this polymorphism to be sure that the consistency polymorph used all over the drug substance lots. They have to provide all the chemical characterization or evaluation, PXRD, TGA or DSC, all the data for at least 3 lots of the drug substances with the polymorph that's consistent. Also, it's recommended to include polymorphic phase testing in the drug substance specification. For the drug product as well, the batches, it should be tested for any change in polymorphism throughout the shelf life of the drug product. This information we are looking for all the time.
Thank you for that response. Again, if anyone else has questions in the room, please come up. But in the meantime, we'll go ahead and go to our next online question.
All right, this question is directed towards Dr. Brian Newman. Recently, the FDA has published PSGs for DPIs that included other alternative in vitro studies that could be used for bioequivalence or product characterization. Can any of these studies be used to test the RLD or a proposed generic?
So that's a good question. I think, so what we can say now is that, of course, what's currently posted in our PSGs are current recommendations, but as was discussed yesterday during the presentations on alternative approaches, we're obviously been looking at both what study designs for alternative approaches will work for both MDIs and DPIs. So I would say that certainly some of these tests can apply for DPIs, and there was a good slide to show that across even with the MDIs, a lot of this depends on the characteristics of the product. So if you're considering this particular DPI, the API itself and its own characteristics, you can try to discern what types of alternative approaches might be applicable for that. So with that in mind, of course, you could then develop a alternative approach and provide that in a pre-ANDA meeting request to get feedback from the agency. And certainly with considering this, I'd also point out that the PSG forecast list provides a good indication of how we plan on working through and considering the different PSGs, which ones needs to be revised for the alternative approaches. So if you're considering developing a product and you have this question in mind, I'd encourage you to take a look at the forecast list to see when we might be providing guidance and consider how you want to develop your program at that point.
Thank you. So this still looks like we're a bit shy in the room, so we'll still move on to our online question. Yep.
This question is directed towards the panel in general. If an API cost per milligram is very expensive for an ANDA applicant, does the FDA accept ANDA applications with smaller batch sizes other than the recommended ones? What is FDA's expectation?
So I'm talking from for example, for the complex drug product, we usually recommend 3 batches on the commercial scale. However, also again, we may accept 1 batch at a large scale and 2 batches, 1/3 of the commercial batch scale. And again, if there is any questions like this, we highly recommend the applicant to use all the FDA communication channels during the development stage through the pre-ANDAs or CC with all the questions, and we are happy usually to help them with any with clarifying any question they have. Okay, thank you.
Thank you. So we'll go on to another online question.
All right, this question is for Tina Zhao. What are the recommendations for the samples used in comparative studies?
Yeah, for the test product, we ask for at least 3 batch of the test product manufactured from at least 2 lot of API tested on or near the at the release and at the end of the shelf life. And also we ask for at least 3 batch of the ROD, I mean, if available, at a different age before the expiry. And also in your submission, we recommend you provide a table to list the test product, the batch number, the API lot number, the manufacturing date, the expiration date, and testing date to help us to figure out the age of the sample. Yeah, that's it. Thank you.
Thank you for that response. Do we have another online question?
Yes, so this could be a follow-up for Tina Zhao and as well as to Dr. Yili Li. If I performed the comparability study aged and near release, it is recommended to update the study with samples aged at the end of the proposed shelf life. But which test needs to be updated? Only impurities profile comparison? Or do we need to also update the aggregate test? Or which tests need to be updated?
Yeah, I think all of them should be updated. Yeah, thank you.
Yeah, I just want to add, the characterization not only for impurities, but also for like high order structure and aggregations. So the study should be tested, should be performed near release and at the end of shelf life. We want to say like at the end of shelf life, your product behave like properly.
Yeah. Thank you again for those responses. And again, reminder to those in the room, this is your chance to come up and ask a question. All right, we've got someone on this side of the room.
I have a question for the panel. So you talk about accelerator testing. And in practical, there's a lot of different rules to use, maybe Arrhenius equation or the 10-degree double rate, those things. But we know that's not universal for everything. What thought accelerator testing you have to unify those things?
Can you repeat the question? Just I missed the
accelerated testing.
Oh, accelerated.
Yeah, accelerated testing for the shelf life. Like it's 2 years shelf life, you do 6 months at higher temperature. And what temperature you're going to choose, what time choose you based on some acceleration equations. Today we use some simple rule like Arrhenius equation or 10 degree for double rate, something like that. My question is not everything follow that equation exactly. And what's general thought to case by case to do that?
I can go on for this question. So I recommend you look at ICHQ1E. it states very clearly what situation, for example, you have, if you fail accelerate, what you're supposed to do is usually you need to start in intermediate stability. And also if intermediate stability increase significantly, what you're supposed to do. So there's a very clear decision tree in ICHQ1E. If you follow that, it will be clearly stated what you're supposed to do. And if the long-term stability is room temperature, what you're supposed to do. However, if it's long-term stability is refrigerated, what you're supposed to do. So I recommend you just go follow the decision tree in ICHQ1E. I answer your question.
Yeah, I agree with Ely. I feel like the guidance is very clear, but I want to bring up one special situation, and this doesn't happen. I have a lot of work done in the PEPFAR space, and sometimes in the PEPFAR space, we have zone 3 and 4. So when dealing with things that we know are going to different climates, such as very humid and, in the Southeast Asia or in the Caribbean or in Africa, in those situations, those are a few deviations that we would say away from the guidance. But in those situations, then they would have a higher, like 30 degrees and really high humidity, and sometimes. So those are the few exceptions, but for the most part, they do follow the guidance in terms of if there is an issue with the accelerated, then you would follow the guidance.
Thank you. And now we have a question on this side of the room.
Hi, it's me again. So you recommended the orthogonal method for the characterization, right, of peptides. So say if we have two methods, we try one of them is not working, like feasibility, it's not feasible. So should we just keep trying till we have a second method, or would that like one method be acceptable in that situation?
So yeah, usually we recommend like orthogonal method. So it's kind of like in cross-checking whether your results are like comparable to each other. Most of the time, so I don't see. Yeah, I think if two methods are not working, you should try more different type of method. And if you really exhausted all the technique and you only found one method working, I encourage you provide like a clear justification for everything, why, like state clearly why this method is not working, what's the problem with that. I would say it will be case by case, but in general, yeah, we do recommend that you have an orthogonal method.
So like say if like for aggregation test, right? Say if one method found no aggregation and then we do a paper-based like a risk justification, the other method, for example, SAC mouse is not working, do you think that can be approach we can take? Very specific, but.
Yeah, it's really like I cannot give you straight answer, like because it depends on the product, also depends on your result. As I said, if you think no aggregation detected with one method, you definitely need to provide a clear information, like whether your result is yeah, whether like your result is correct, so you definitely need to check. And I don't see, yeah, so for peptide, usually we recommend you do like high order structure also. Actually, if you have aggregates, sometimes NMR is can also be a good indicator, like whether there's aggregate. So NMR, high resolution, like two-dimensional NMR can like double check both the highest order structure and the aggregates, whether there's an indication. So I don't say like, I mean, so far I don't say like none of the method working for certain thing.
Thank you.
All right, we have one more question here.
Yes, I'd like to know, will the agency grant 36-month dating on an initial application, an ANDA, for a complex product, if you have the 36-month data for both your product as well as the brand. And just to be clear, the size of the manufacturing that you've submitted is the size that commercial will be at. It's the commercial scale.
So your question is, if the applicant provided 30-month stability data for his product and also the brand in the commercial scale, in the commercial manufacturing site?
Yes.
What the question in this case?
The question is, would you grant 36-month expiration date?
It depends on the data. If the applicant propose 30 months shelf life, we will work on that to see if it's acceptable or not based on the data provided. If the applicant provided 24 months and they have 36 months, but they propose 24 months, we'll not tell them, yeah, you have until 36, you can extend it now. No, it's the applicant decision. But the data provided based on what the applicant provided, what he is proposing. You having your application in P8, you're proposing 24 months shelf life. This is our shelf life. Extra data, we review it for sure, and if it's adequate, but we accept 24 months based on the proposal provided to us.
Right. Okay. So you can, if you have 36-month commercial scale, it seems the agency would take that into consideration. And we
It's reviewed.
propose.
Everything is reviewed,
Yeah.
but at the end, the product approved based on the proposed by the applicant. Applicant proposed 24 months, that's the shelf life. Yes, if it's everything is fine, it's fine. If applicant would like post-approval, they can provide a request to extend the shelf life or whatever based on the data already provided. Or if they feel that the product already up to 36 months adequate data within the specifications, they can request to extend the shelf life to 36 months.
Okay,
It's up to
thank
the
you.
applicant. Okay.
All right, thank you so much. Looks like that's all the time we have questions for, or all the questions we have time for. Thank you again to our presenters and panelists for all the excellent information and insights. We'll now be taking a break which will resume at 11 a.m. Eastern Time.